Hi everybody, this is Mrs. Ching. This video is to walk you through how to complete the multiple alleles problems, such as the ones you will find on problem set number 4. Okay, so this, in order, if you need to have a reference, this is going to be in your book under chapter 14.1. Okay, so the one trait or one prime example for multiple alleles that we see is human blood type. And that's the one that we're going to really focus on in this section. Okay, so you can see that there are many different blood type here, such as blood type O, blood type A positive, A negative, B positive. I'm actually A B positive, which only 4% of the people in the population are. And this is only in the US, so it does vary differently in the world. Okay. So what is going on with multiple alleles and how does that explain blood typing? So you have a diagram that looks like this on your problem set 4. So this is going to be in your problem set number 4. Okay, so let me explain this slide a little bit more for you. What's going on here is these are the 4 blood type, right? A, B, and O. So the genotypes that we're going to be using, or that we are really looking at here, actually, let's define the alleles first. The alleles that we're dealing with are going to be I with A in the superscript, I with the B in the superscript, or little I. So IA and IB are both dominant. These are both dominant. And they show up, they act a lot like the codominance trait that we've talked about, like in the roan cows. This little I is going to act like the recessive allele. Okay, so what does that all mean? So let's look at this chart here, this table. So you can have 2 different genotypes for blood type A. What that means is if you have the allele for 1A or IA, you're making this antigen. So this carbohydrate that's listed here is really an antigen. Antigen A is being made. If you have blood type B, you have antigen B. Antigen is a surface protein or surface carbohydrate rather that's expressed on the surface of red blood cells. So if you're AB like me, I am making my red blood cells have both A and B. If you're blood type O, there's no antigens. Okay, so then the big worry when you donate blood or receive blood is you have to match up the blood type. But you can see here in this diagram, there's many different things that you got to look at. Okay, so for example, I like to use my example because I am actually the universal receiver. I can take anybody's blood because I can take, because I have both A and B, I can take blood type A here and I would be fine. So this is what it would look like if I were to receive blood type A. If I receive blood type B, I'm also fine. But my dad has blood type B. And if he takes in my mom's blood, which is blood type A, if he gets a transfusion from my mom, this is what's going to happen. It's going to coagulate. It's going to clump up, and then we'll have issues, right? So that's not good. Okay, so that's what these antigens mean. These little alleles code for antigens. The little I means there's no surface antigens at all. Okay, so these are the different genotypes that you're going to want to refer back to this when you do the problem set. Okay, so we are going to then approach the first problem. Okay, so here's your first problem set. Again, this there's a table in here for you. So first thing you got to do is figure out what the heterozygous A stands for. So remember, heterozygous means different allele. So heterozygous A is going to look like this and like this, just like in this example right here. Okay? So then you're going to cross this with a person that has AB. So it's going to, whoops, sorry. It's going to look like this. Okay? Then you do your Punnett square. Just like how we've always done it. Do make sure that you're writing your letter A and B as a superscript, slightly higher than the rest, because I don't want you to get confused that that's the second allele. It's not. It's attached to that I. Okay? Okay, so there's your 4 possibilities, and then you need to then summarize the results. So I basically did step 2, 3, and 4 together here, and then step 5 is going to be over here. Okay? 1 I didn't have to do because it's already written out here for you. Okay, so I'm going to delete that. Okay, so what we're going to do now is summarize the result. So you have, whoopsies, uh-oh. Okay, so we're going to summarize the result. 25% will be this, homozygous for A. And then you're going to have 25% IA, IB, 25% IA little I, 25% IB little I. And then don't forget to do your phenotypes correspondingly. So you can refer back to this table to figure out the phenotype for all of these. So this would be A, AB, A, and B. Okay, so that's the first problem. Double check on the key if you need additional help, or just save your questions. See me after school, or save your questions and come or talk to me next class. All right, good luck.